Okay, welcome to the first breakout session. I'm Casey Luskin, Associate Director of the Center for Science and Culture here at Discovery Institute. Hope you guys are enjoying the conference so far. Okay, awesome. I'm looking forward to this talk a lot because Ray Bohlin has been a longtime friend of mine. In fact, Ray, we first met back around the late 90s when I shared a tent with his son on a mission trip to Costa Rica, and had no idea who Ray was at that time or that someday I'd be working with him. But Ray is a wonderful person, and you guys are really going to enjoy this talk. He earned his PhD in molecular and cell biology from the University of Texas at Dallas. He's currently Vice President of Vision Outreach for Probe Ministries, and he's also a longtime fellow at Discovery Institute. How long have you been a fellow at Discovery, Ray? It's been
since '98.
Since '98, so pretty much since the beginning, almost since the beginning of the Center for Science and Culture. Ray has lectured all over the world on numerous college and university campuses. In fact, another interaction I had with Ray was about 3 years ago. We hosted him in South Africa when he went on a speaking tour of South Africa while I was living there. We had some fun times. And he has published in a number of journals, including the Journal of Thermal Biology, Journal of American Scientific Affiliation, the Journal of Mammalogy, and he is the author of the book, The Natural Limits to Biological Change, which you may not be aware of this, but that is one of the founding books of the intelligent design movement. And so Ray is going to speak to you all today about the topic of the natural limits of biological change and help us understand where this topic stands today. So Ray, thank you so much.
All right.
I don't do well holding a microphone while I'm trying to talk. I'm a one thing at a time kind of person, so I'll move it a little bit closer. Do you really need the microphone to hear me? Yes. You do? Okay. I said, do you need the microphone to hear me? Okay. This book was published in 1984, initially. My co-author was Lane Lester, who at the time was the PhD of the two of us. A PhD in genetics, and we needed somebody like that at Probe Ministries. This was part of a book series that we were publishing at the time, and this was the original. It was part of what was called the Christian Free University curriculum. So it was books from a Christian point of view, where the Christian point of view was, I could say it was underneath. It was latent within the material, but it wasn't overtly Christian in any way. Okay. And Lane and I got together to put the basic outline of the book together, and then I came back to Dallas, and I wrote the book. Okay? And he did an index and that sort of thing, but of course he proofread some of it. Most of what he did was copy editing, and it wasn't until about 10 years ago. He may even be deceased by now, but I'm not sure. He apologized to me that he's gotten all kinds of kudos for this book over the years, but you know what? You really wrote it, and I never told anybody that. So anyway. The book currently, which you can find in the bookstore area, is this cover, okay? And initially this book series was published through Zondervan Press, and then we ended up taking it over. It's now published, but was then published by Probe Books. Probe no longer publishes books, so it's a bit of a dinosaur in a lot of different ways. But Discovery is very interested in me doing a revised and expanded edition. There's lots that's happened in the last almost 40 years. And we have a tentative target date of somewhere in 2024, which would be 40 years after the original was published. So I'm going to just give you a quick overview of the various sections, okay? First of all, there they all are. They're supposed to come up one at a time. Anyway, just a brief couple of pages about the nature of biological change in general, what we know about it at the time. And I talked about the variety and complexity of life. I just gave a lot of different examples about the various different kinds of amazing complex biological forms that we have. One of those that I definitely want to include is something called the mantis shrimp. Anybody know what a mantis shrimp is? Heard of it? Yeah, they're pretty amazing in that we have 3 different kinds of cones to detect color: blue, green, and red. The mantis shrimp has 8 different cones for different types of wavelengths of light. Why in the world they need that? Nobody has any idea how or why. What's the point? Okay, they live under the water. Colors are kind of muted, right? Anyway, that's what I'll be adding. When I talked about genetics in general, this book was dedicated to someone who'd been in college and maybe had at least one genetics class. So I did give a quick overview. Then the book came in two different sections. Three sections. First one was about neo-Darwinism, which is primarily still what we're talking about today. So I explained it as best I could objectively. I had just completed my master's degree here at the University of North Texas, which was in population genetics. And today that would likely be called evolutionary biology. And I reasoned if I wanted to be a critic of this idea for the rest of my life, I better make sure I know how they say this is supposed to work. So I emerged a greater critic of evolution when I left than when I went in. Then the next chapter was critiquing that. Neo-Darwinism under attack. The next section was about what then was called punctuated equilibrium. Not as popular today for a lot of different reasons, which I'll talk briefly about. And then I offered some obstacles to that as well. Stephen Jay Gould and Niles Eldredge were two primary individuals who promoted this punctuated equilibrium idea. We'll talk about it a little bit later. And I was very gratified when I wrote him to ask for permission to use a figure from one of his publications. And I gave him the chapter on evolution by speciation. I provided the chapter on the obstacles to it so he could know what this was going to fit into. Well, he could tell I was a creationist, right? So he wrote back and said, well, it's not my policy to give permission for such a thing, but based on the book, the chapters you showed me, you can't just do what others have done. You redraw the figure and just say it in the note after this is a figure. He told me to do that. I mean, I thought that was a good plug, if you will. And then the last chapter was simply called Another Alternative, taking all of these objections to Darwinism, to punctuated equilibrium, and bringing them together in a new view. This is one of the first books where the phrase intelligent design was ever used. Intelligent design really didn't get started until about the mid-90s after Behe's book, Darwin's Black Box. Phil Johnson, with his book, Darwin on Trial, in 1990, kind of started the ball rolling. But ID really wasn't a thing until the mid-90s or so. And so what we're concerned about here, evolution, at least at that time, is the common descent of all organisms by the process of mutation, natural selection, is assumed to be able to explain everything we see in the biological realm. I'm also, as Casey mentioned, a speaker and a writer, researcher for Probe Ministries. It's a worldview and apologetics ministry out of Plano, Texas. And this is really much more of a worldview question than it is a scientific question. What are you coming to the data with? If you're coming with a naturalistic worldview to start with, you'll interpret things naturalistically. If you're coming at it with a theistic worldview, you're going to interpret things, again, very differently. Okay? Richard Dawkins has become one of my favorite authors for lots of different reasons. A book published after Natural Limits, he said this on page 1, biology is the study of complicated things that give the appearance of having been designed for a purpose. They just look designed, okay? They're not. He spends the rest of the 300 pages of his book explaining how they're not designed, okay? There's also another quote I sometimes use by Francis Crick. It says, where biologists must constantly keep in mind that what they're observing was not designed, but rather evolved. If you have to keep telling yourself this is not designed, that should tell you something. Okay? So, well, there are a couple of things that challenge this idea. This idea of convergence. That's where different groups of organisms arrive at similar solutions to similar problems. For instance, in the difference between placental mammals and marsupial mammals, there's a rat-like organism in both of these. There are grazers, kangaroos and koalas, cattle and others among placental mammals. There's carnivores with marsupial mammals. There's carnivores with placental mammals. And they arrive at very similar conclusions, solutions, I should say. Then you have these different kinds of ways of getting around. If you're moving through the air, you need some kind of wing, whether it's an insect wing or a bird wing or a bat's wing. If you need to move through the water, you need some kind of fin, whether that's on a bony fish, like a largemouth bass, or whether it's on a cartilaginous fish, like the shark, or whether it's mammals, whales, orcas, dolphins, porpoises. They all need fins of some kind to help guide them through the water. So, if we're going to rely on evolution, these same kinds of solutions have been hit upon again and again and again. There's only so many different ways to move around in water. Appendages for walking, whether you're an insect with 6 legs or a human with 2 legs, or you're a reptile, again with 4 legs. There's only so many different ways you can get around on land if you're actually going to walk, okay? And the appendages are analogous. In other words, they're accomplishing the same thing, but they're not evolutionary related at all. Organisms have arrived again at a similar solution. So what we can see here is that there are definitely physical constraints to the kind of biological change that occur. The question then becomes, what about biological constraints? Are there biological constraints on the type of change that can take place? So change is not limitless. So what could be the limits? Well, one of the first things to remember, if you know Steve Meyer's book, Darwin's Doubt, and the film Darwin's Dilemma, the stunning burst of body plans in the early Cambrian and the lack of significant new body plans since indicates there's a limit to change. In 1995, Time magazine published an article that was the cover story, and within that, one of the developmental biologists who was working on this problem said an amazing thing. There must be limits to change. I read that and said, oh my gosh, an evolutionist just said that. That's the title of my book. After all, we've had these same old body plans for 500 million years. So there's even a recognition to some degree within the biological community that there are limits to how much change is going to take place. Now, what we're going to look at here are several fairly common examples of natural selection. Artificial selection, the peppered moth, and what are known as the Galapagos finches or Darwin's finches. What is natural selection actually able to accomplish? Well, with artificial selection, that's been used as an example of the power of natural selection ever since Darwin's Origin of Species, one of the primary examples he used. Now, we've done a lot of artificial selection. Roses, dogs, pigeons, horses, cattle, the protein content in corn, the sugar content in beets. All those and many, many others have been highly selected. But eventually, the quality fizzles out. You can only get so much protein in an ear of corn, or in a kernel of corn. You can only get so much sugar into a beet. And the same thing with some of these others. One of the things Darwin played around with was domesticated pigeons and breeding pigeons, and how many different varieties of pigeons can he come up with. Anybody know what the real difference, though, between artificial selection and natural selection is?
Intelligence.
Intelligence, yes. With artificial selection, there's a mind that wants to achieve a particular result and starts breeding things intentionally to accentuate that particular trait. Now, a little clutch of eggs here. This is a quote from Natural Limits. However, chickens don't produce cylindrical eggs. We can't produce a plum the size of a pea or a grapefruit. There are limits to how far we can go. Some people grow as tall as 7 feet, some grow no taller than 3. But there's nobody over 12, nobody less than 2 feet. Even within a particular form, the limits are pretty well set. You can't get much beyond this one, you can't get much beyond on the lower end as well. When you come to the peppered moth, this is really fascinating for me, it has been for a long time. The peppered moth story begins in the 1950s. And it was the first example they came across of Natural Selection in a Wild Population. Darwin published in 1859. It took them almost 100 years to find a good example of real natural selection. Okay? So again, one of the earliest examples. Now, what began happening, you can see from on your right side here, that in the upper picture, there's two different forms of the moth. The one on the upper left is the peppered variety. The one on the lower right is called the melanic form. So those two forms basically always existed in the population. And ordinarily, before the Industrial Revolution, the trees in England were fairly light-colored beech trees. They had lichens growing on them. And the peppered variety was easily conspicuous, was easily camouflaged. The melanic form was easily seen. When the Industrial Revolution came on and lichens started to die and the trees started getting coated with soot, well, suddenly the peppered variety was easy to see and the melanic form was hidden. And what happened was prior, the peppered form far outnumbered the melanic form, 99 to 1 or more, okay? And then after the Industrial Revolution, the frequencies flipped. So that now the melanic form became more dominant and the peppered form less so. Well, all that happened was a change in frequencies of the two different kinds of moths that already existed. What people thought was that bird predation must be the mechanism of selection because we're talking about the visibility of these moths on different kinds of background. Now, they ended up finding out a lot of things here, which I don't have time to go into all of this. However, the main thing here is that the genetics of what's called industrial melanism is not known. We don't know what the genetic difference is between the melanic form and the peppered form. Both forms, as I said earlier, appear to have always existed. The frequency simply flipped. And the mechanism of selection is also in doubt. The only consistent correlate, we found industrial melanism in other populations of moths in different parts of the world. The only corollary seems to be that pollution was present. Not predation by birds, not the death of lichens, none of that, just pollution. Sometimes there were no birds predating on these moths. Sometimes the lichens were just fine, but pollution was present. How that relates, nobody knows, even to this day. Then we have the example of Darwin's finches. By the way, I've had the privilege of visiting the Galapagos Islands. I was there for about 4 days. And what you see here are 11 of the 13 forms of Darwin's finches. And across the top 2 rows are all referred to as ground finches. So you have the small, medium, and large ground finch. You have the sharp-beaked finch. And the 2 cactus finches are also ground finches. And then there's a couple of tree finches. There's actually 4. They only picture 2. There's a vegetarian finch, and then a woodpecker finch that actually picks up a little stick and digs around for insects to pull them out with a stick, like a woodpecker would. And then there's the warbler finch. Okay. Now, what's interesting here is that Peter and Rosemary Grant have been studying these since the early 70s. They've returned to the Galapagos Islands for 3 to 6 months every year since. I think they're both retired now, but I think they still go back. And they came to an island called Daphne Major, and it's several miles away from the main island area, and so it was fairly isolated. Birds could fly from back and forth between the islands, but not very much. And so they began charting, measuring the size of the beaks, the type of seeds they were eating. They were able to observe almost everything. And if you don't know much about the Galapagos Islands, all of the animals that are endemic or native to the Galapagos Islands have no fear of humans. You could walk, these finches, if you were carrying a pole and you just kind of walked around like this, they would start alighting themselves on the pole that you're carrying. They come in and sit on your head. Or if you're standing still, they come start poking around your feet. Okay? So very friendly, easy to capture and easy to measure, and their weight, the size of the beak, all sorts of things. So they've done this for a very long time. Now what we've learned is that there's both a wet and a dry season in Galapagos, but there's also a period of time where there could be several really dry years in a row, and then there can be several wet years in a row. The wet years almost everything blooms. There's all kinds of seeds, from the very small to the very large. What happens when you go through the several year period of a wet season, there's an abundance of these smaller seeds, and the beak size of almost all of these ground finches gets a little bit smaller, by tenths of a millimeter. How'd you like to get to measure that? And when you get to the dry season, all these small seeds start disappearing. And over those several years, the beaks all start getting larger for all of these species. So there's a continual fluctuation back and forth. We've also measured their mitochondrial DNA. The mitochondria in your cells are the powerhouses where all your energy for the cell is made. And it's a small piece of DNA, usually has about anywhere from 15 to 20 genes on it. That's about it. It's a circular piece of DNA. And we've sequenced the mitochondrial DNA for all these different finches. Now look at the very top. Those are the 6 ground finches. They're basically indistinguishable from each other with the mitochondrial DNA. When you move down below and you see the tree finches, well, they're a little bit more distinguishable, but not by much. In other words, what we're seeing is that there's lots of gene flow between all these different supposed species. Now, the ground finch species do interbreed at a frequency of 5%, maybe a little bit less, but at least 5%, around that level. Tree finches, we know, do also interbreed, but their frequency and fertility has not really been established. And the ground finches, as I just showed you, are not separable by mitochondrial DNA, and tree finches only somewhat more so. We do know that beak size is heritable, but at the time the specifics were not known. We now know a little bit more about that. But the point here is that many are now starting to consider these 6 ground finch species as one species. And each of these is simply a subspecies or a race of the one ground finch. Could be the same thing with the tree finches as well. So there has been some adaptive radiation. One population of these birds arrived on these islands, they suspect, in an evolutionary time frame about 2+ million years ago. And then with all the different food sources available, some simply started specializing on small, medium, large seeds, or getting into trees, actually eating foliage, which birds don't ordinarily do, but somehow these finches started to do that. And so there was a radiation, if you will, but it's still in progress. It's still fluctuating back and forth. Now, peppered moth, Darwin's finches, excellent evidence for natural selection acting upon variation that's already present in the population. We can't go back in time to trace this, but it is suspected almost all the variation in these finches was present in that population of finches that was blown over from South America, or the Caribbean, they actually think, to these Galapagos Islands. All that variation was already there. It simply got sorted out over time. And no one, as far as I know, quibbles with natural selection as a real phenomenon in nature. The question then becomes, what does it really tell us about evolution? If it's only really acting upon variation already present in the population, it's not really telling us that much by itself. So this is where the idea of mutation has to come in. So natural mutation plus natural selection. There has to be a source of variation. Darwin didn't know what it was at the time, but now we understand that it's the mutations that give rise to the so-called variations that natural selection that can then act upon. The mutations are categorized in 2 different ways, what are called structural genes and what are called developmental genes. So structural gene mutations, I'll define these as which code for a protein which performs basically a maintenance, support, or some kind of specialized function. Just because it's called a structural gene or protein doesn't mean it actually forms the outline of the cell. It could be an enzyme function, it could be a maintenance function within the cell. Lots of different enzymes are structural genes. Developmental genes, however, influence specific tasks in embryological development, or sometimes they were called initially regulatory genes. Now, most examples of mutations that we have documented involved in speciation or supposed evolutionary change have always involved just structural genes. These tend to be genes that have their expression fairly late in development. They're not changing the overall structure of the organism very much, but they are providing slightly different capabilities than what the organism already had, such as microbial antibiotic resistance. Sometimes there's already a process in place by which bacteria will pump the antibiotic out. It simply gets better. It gets more efficient. There's also a way already in place in bacteria to deactivate the antibiotic. Again, it just becomes a bit more efficient. Sometimes some antibiotics target what's called the ribosome, where protein synthesis happens. And so sometimes there's a change in the ribosome such that the antibiotic doesn't interact with it very well. So the organism is able to survive in the presence of the antibiotic. The mechanisms of resistance can be achieved naturally by mutation or acquired through gene transfer. That's how most other species get antibiotic resistance. They transfer the genes from one organism to another. They don't create any new mutations. But even with the target molecule, what always happens here is that there's always a metabolic cost. Yes, these bacteria can survive in the presence of the antibiotic, but you put them in the presence of the wild-type bacteria, take the antibiotic away, and the wild-type outperforms and they disappear. Antibiotic-resistant forms always disappear. When you affect the ribosome, you make it a less well-functioning ribosome. They can't keep up the pace of the normal bacteria. If you've read Behe's latest book, Darwin Evolves, you'll be familiar with that. So antibiotic resistance really doesn't involve the creation of a new or novel function in the cell. These are things that already existed. They were just improved or made more efficient. Antibiotics exist naturally. Where'd the first antibiotic come from? What's it called? Remember? Penicillin. Where'd we get it from? Did we make it? Penicillium. It comes from a mold. So the penicillium was already protecting itself against bacteria using this molecule. So it already existed, so it kind of makes sense, would you think, that some bacteria would have an ability to resist that antibiotic. Basically, no new genetic information is required. So we're beginning to see, okay, there are some changes happening, but they're not really involving much that's actually very specific, or actually nothing new has actually arrived. We're just rearranging what's already present. I use this quote in the book from Pierre-Paul Grasset, who at the time was the dean of French evolutionists. He said, "What's the use of their unceasing mutations if they do not change? In the sun, the mutations of bacteria and viruses are merely hereditary fluctuations around a median position. A swing to the right, a swing to the left, but no final evolutionary effect." Now I'm talking about this difference between what's called micro and macroevolution. These are not terms invented by creationists. These are terms that are used in the evolutionary literature. Sean Carroll simply said that a long-standing issue is whether the processes observable in extant populations, meaning microevolution, are sufficient to account for larger scale changes evident over long periods of time. Now, what he's simply saying here is we're not really sure if these micro changes can add up to a macro change. Andrew Simons, 2002, put it this way: persistent debate in evolutionary biology is one over the continuity of microevolution and macroevolution, whether macroevolutionary trends are governed by the principles of microevolution. In other words, there was a lot of doubt as to whether microevolution leads to macroevolution. They weren't convinced. That became a bit more clear recently. Scott Gilbert, therefore, to study large changes in evolution, biologists needed to look for changes in the regulatory genes that make the embryo, not just in the structural genes that provide fitness within populations. So, the primary need for macroevolution, what you need is some new arrangement of the various parts. Oftentimes this is referred to as a body plan, or the technical term is Bohlin, B-A-U-P-L-A-N. So what is a body plan? Well, what you see above you there, those are arthropods. insects, crustaceans, centipedes. Their body plan is where the skeleton is on the outside of the organism. It's an exoskeleton. Well, then you have the chordates, fish, amphibians, reptiles, birds, mammals. Their skeleton's on the inside of the organism. Now, somewhere these two phyla have a common ancestor. But how did one of them get a skeleton on the outside and the other one get a skeleton on the inside? You have to change things from the very beginning of embryological development in order to bring about that kind of different body plan. Now, a book by Wallace Arthur came out in '97, and he was explaining that structural genes tend to have little effect on the development of the body plan. So enzymes and building block proteins don't have an effect on the body plan. The developmental genes that do affect body plan tend to find their expression quite early in development, not later, early in development. And here's the quote. This is a book I found during my first fellowship back in '98. Those genes that control early developmental processes are involved in the establishment of the basic body plan. Mutations in these genes will usually be extremely disadvantageous, and it's conceivable that they are always so. As Jonathan Wells had said, when you have an early development and mutation take place, you either get the same organism, a defective organism, or a dead organism. There's no other option. Playing with these early development mutations does not bring about any kind of positive result. And I love this by Gilbert, Opitz, and Rath. The modern synthesis, meaning neo-Darwinism, is a remarkable achievement. However, starting in the 1970s, many biologists began questioning its adequacy in explaining evolution. This has been an oft-repeated phrase here. Microevolution looks at adaptations that concern only the survival of the fittest, not the arrival of the fittest. Very, very different things. How organisms survive, well, that's microevolution. How they might change, responding to a small little change in that particular environment. But to get a new type of organism, that's the arrival of something different. And neo-Darwinism doesn't really answer that. Wallace Arthur, again, said in a developmentally explicit approach, it's clear that many late changes cannot accumulate to give an early one. Thus, if taxonomically distant organisms differ right back to their earlier embryogenesis, as is often the case, and I'll say is always the case, the mutations involved in their evolutionary divergence did not involve the same genes as those involved in a typical speciation event. What evolutionary biologists have been studying for decades does not involve macroevolution. It simply doesn't. It gives you, again, that impression. Are there limits to change? And if you're going to break out of those limits, what's going to be required? When we've tried to do that by mutating developmental genes, it's disastrous. So how can you change body plans? So what do we have so far? Significant morphological change requires early developmental mutations, but these mutations are nearly universally disadvantageous. Therefore, this would be expected to be a slow and difficult yet bumpy process. We don't see that in the fossil record. Indeed, Darwin expected that as natural selection acts solely by accumulating slight, successive, favorable variations, it can produce no great or sudden modifications. It can only act at short and slow steps. All of our experience is telling us, however, that when you change any developmental gene, you either kill it, or it's extremely disadvantageous, or you get a normal organism. So you're not able to change. If you try to accumulate small changes, you just get the same organism. There are no small early developmental changes. I know I'm talking fast. I'm not talking as fast as Casey does, so hopefully you're able to follow a little better. This is the Time magazine article I was referring to. This was just after the Shang-Jane fossils were found in China. And previously, the primary site for Cambrian fossils was the Burgess Shale in Canada, and the Shang-Jane were about 5 to 10 million years earlier than that. Now, Simon Conway Morris, who, by the way, is a theistic evolutionist, he is a Christian, the term explosion, he said, should not be taken too literally, but in terms of evolution, is still very dramatic. What it means is rapid diversification of animal life. Rapid in this case means a few million years rather than the tens or even hundreds of millions of years that are more typical. In other words, to get a new phylum, a new body plan, they would have expected that to take at least 10 to 100 million years. But here in the Cambrian, we're seeing that kind of change in 5 to 10 million years. Now, a 10 million year window with radiometric dating, that's about as precise as you can narrow the time frame down. You can't narrow it any further than that. So that's why some say 5 to 10. Somewhere in that 10 million year window is the Cambrian explosion. Now, prior to this, what's 22.1 billion years ago, according to evolutionary time frame, to 620 million years ago, little else but prokaryotes, bacteria, cyanobacteria, some eukaryotic algae, maybe. Then the period called the Vendian, 620 to 550, sponges, some cnidarians, some flatworms, the platyhelminths, and maybe some annelids, or segmented worms like the earthworm. However, both, especially with the annelids, what we found were little like wells dug into sediment, but there was no organism in them. So it's what's simply called a trace fossil. Maybe a worm made that hole, maybe it didn't. But we do find things that look like jellyfish or cnidarians. We do find examples of sponge parts like spicules and some actual things that look like sponges. And within that Vendian is something called the Ediacaran fauna, which is named after a location in Australia. And they were a very different group of organisms. Now, I'm not going to go into much detail here, but you see on the very right-hand side the Ediacaran biota, and you see them indicating 5 different basic types that you could find. And these are all very strange organisms. There was no skeleton of any kind, exo or internal. Fairly flabby things like fronds that just kind of waved. Some thought they were plants for a while, but then fairly certain that they were animals. But then they all went extinct, just prior to the Cambrian. They all disappeared. And the Cambrian animals, for the most part, say they have no resemblance to the Ediacaran fossils. They existed for a while, they go extinct, and then shortly after comes the Cambrian explosion. Now, the Cambrian period is about 550 to 485 million years ago, and you definitely add a very specific flatworms, arthropods, mollusks, segmented worms, annelids, chordates, echinoderms, like starfish. These all very clearly show up. All these different animal phyla show up with no warning, and we didn't, where they came from, how they're related to each other, we have no idea. Actual explosion occurs, as I said earlier, between 545 and 535 million years ago. So when before, most of what you had was prokaryotes and cyanobacteria and some of those Ediacaran stuff, suddenly you see this. A whole different grouping of organisms. Basically now what we believe is essentially all of the animal phyla have their beginning in the Cambrian period. There are some phyla that contain organisms so small they don't show up anywhere in the fossil record. We have no fossils of them at all. So some are suspecting they actually almost all do show up in the Cambrian. And I like this quote from that same Time article by Samuel Bowring, who was a geophysicist who helped narrow down that 5 to 10 million year period. He said, "We now know how fast is. What I like to ask my biologist friends is, how fast can evolution get before they start feeling uncomfortable?" It ain't supposed to be that fast. Usually the speciation event is something they say is 1 to 2 million years. But now you've got this whole different body plan showing up in just a little bit more time than that. Paul Chen, in a recent book from Discovery Institute Press, talks about the Cambrian Explosion book by Irwin and Valentine. In that book, the authors remain committed to searching out a purely material evolutionary account of the Cambrian explosion. And yet they insist that the singular event in the history of life remains marked by important, I love it, unresolved questions. And they call the transition from sponges in the Vendian to the creatures of the Cambrian the most enigmatic of any evolutionary transition in metazoans. Metazoan simply is multicellular animal. They're committed to finding a naturalistic explanation, but they don't have one. And it's the biggest mystery there continues to be. So again, are we seeing a limit to biological change being expressed here? So what do we have? Well, neodarwinian change is slow and gradual. Body plans are expressed early in embryological development. Mutations there are nearly always disadvantageous. Yet in the Cambrian shows numerous new body plans in an extremely short period of time. How can that happen naturalistically? Then there's the rate at which mutations occur. This is also now a major difficulty. Again, 96, Susumu Ono, a Japanese geneticist, said it still takes 10 million years to undergo 1% change in DNA-based sequences. Humans have 3 billion base pairs of DNA. A 1% change, 30 million base pairs. Still takes 10 million years to get that kind of change. So the emergence of nearly all the extant phyla of the kingdom Animalia within the time span of 6 to 10 million years can't possibly be explained by mutational divergence of individual gene functions. He's just flat out, it can't happen. We don't know any way naturalistically that this kind of change could happen by just changing individual gene functions. Now, punctual equilibrium. The basic idea behind this is that species remain unchanged for most of their history. So in the fossil record, a species is found, it stays the same for a couple million years or so, and it disappears. Something new replaces it, okay? So that's what's called the equilibrium portion, or what they termed as stasis. And amongst individuals like this of Gould and Eldridge, stasis is the primary evidence of this. That's what can easily be observed in the fossil record. We find species existed for several million years and suddenly disappear, and they're replaced by a slightly different type of species of the same form, same type. And then the equilibrium is then punctuated by a relatively rapid change to a new species. They introduced the term species selection. And that was a new concept. Selection was always termed as acting on the individual. Either it survives and reproduces or it does not. But now they were suggesting that species can actually be selected. So it was always more an observation of the fossil record. Few intermediates between the species. Like I said, the old species disappears, a new one suddenly appears. The problem, however, was always the mechanism of rapid speciation. And Gould and Eldridge came out with this idea in about 1973. 20 years later, in 1993, they published a review article, and it was titled "Punctuated Equilibrium Comes of Age" in the journal Nature. It had one very interesting sentence in it. Continuing unhappiness. Scientists are unhappy? That's just a weird way to put it. But continuing unhappiness, justified this time, focuses upon the claim that speciation causes significant biological change. For no validation of such a position has emerged. Biologists didn't like punctual equilibrium from the start. What possible biological mechanism can bring about rapid morphological change in a speciation event? And it still seems to be the case. We're just going to skip through this one. We're running out of time. I just want to spend a little time. This is the last thing here. You know what woodpeckers are, right? Woodpeckers are very strange creatures. They bang their heads in the trees. Right? And why do they do that? Find bugs. They will either flip the bark off and search for bugs underneath the bark, or they drill a hole and they want to expose little tunnels, okay, that the insects have made or the larvae have made, and they slip their tongue in and search up and down the tunnel, and they got a very sticky saliva, and they just pull them out, and that's breakfast. Well, woodpeckers have a number of adaptations that allow them to do this. First of all, the bill is very tough. It's not brittle, it won't break, it's not rubbery, it doesn't bend. You see how the tail pushes up against the tree. That acts like the third leg of a tripod. Most birds have 3 toes in the front, 1 in the back to grasp a perch. Woodpeckers have 2 toes in the front, 2 toes in the back. to grasp the surface of a vertical surface. And we've now determined that a woodpecker generates about 1,000 Gs of force each time it hits the tree. Now, a concussion in humans is when the head receives about 10 Gs, all right? And your football player, concussion protocols, all kinds of things. You can die. They just keep going on pecking away. It's no big deal, right? How in the world do they do that? Well, there's some very ingenious things going on. Yes?
Don't they have doesn't their tongue wrap around the back of their skull?
Yeah, I'm getting there. A little ahead of me. This is just fun, okay? I'm a biologist. I'm a critter guy. My tie is a eagle. I wear critter ties. Yeah, anyway. This example was in the original Natural Limits, okay? Well, the bill has an upper portion, lower portion. The lower portion actually sticks out a little farther than the upper bill does. So that's what makes first contact with the tree. And so that pushes that lower bill back, and a lot of the initial force gets forced down through the vertebral column and out through the tail back into the tree. We get concussions because we have fluid between the skull and the brain. And a concussion happens when you have a force and that brain pushes the fluid aside and you get a bruise on your brain. With woodpeckers, there is no fluid between the skull and the brain. So the brain does not move when it hits the tree. There's also a shock absorber of cartilage and muscle at the back of the bill in front of the skull. A lot of different things happening to protect the woodpecker when it does this. But that's not the most unusual thing about the woodpecker. As was just mentioned, they have a very strange tongue. And this illustration is in the original Natural Limits. What you see here is that you see the very tongue tip, okay, in the middle of the page, and then it goes into the bill, makes a loop down into the throat, comes up over the top of the head, down between the eyes, and inserts into the right nostril. So this is going to show you the path it takes from the tip of the tongue. It's all between the skin and the skull. Now move over to the other. Between the eyes, inserts into the right nostril. And if you don't believe me, there's a bone inside the woodpecker tongue called the hyoid bone. And here's a skull of a woodpecker. The problem here is evolution has to accomplish two things at the same time. Never very good at that. You have to evolve a longer tongue and a retraction mechanism at the same time. Can't have one without the other. Doesn't make any sense. So as Stephen Jay Gould put this once in one of his early books, he referred to the dung mimicking insect. He says, well protected, but can there be any edge in looking only 5% like a turd? The problem is always at the start of things. If you're changing slowly and gradually to a woodpecker tongue, the early stage, if it's a slightly longer tongue, maybe a slight retraction mechanism, doesn't do anything for the bird. It accomplishes nothing. There's nothing to select for. So this is one of the observations he made about Darwinian evolution, that something's not right here. Okay. So the quick summary here, only 5 minutes over. How's that? How about that? Artificial natural selection tell us little about the origin of novelty. Mutations, whether structural genes or developmental genes, also offer little help in understanding the origin of novelty. Oops, I'll put that back up there. So, things I've just talked about here, the selection, the mutations, what kind of mutation it is, it's not telling us where biological novelty comes from. Now, new material I'm going to be researching is more about evolutionary developmental biology, called Evo-Devo. And that's really more about, again, the developmental changes that are needed to bring about significant evolutionary change. Something called orphan genes. As we have sequenced more and more animals' genomes, we're finding sequences that are referred to as open reading frames, or ORF, ORF. Okay? But the problem is, if we assume that that's coding for a protein, and we look at that, the sequence of that gene, we don't find a gene in any other kinds of organisms that match this gene. It's an orphan. It's by itself. It has no ancestors. Now we're finding, even with humans, there are literally dozens of suspected orphan genes. That there is no copy similar in chimpanzees or gorillas or any other primate. Where did these brand new genes come from? And how do they arise by any sort of mutation, natural selection? Need to check back up on the status of punctual equilibrium. Gene and protein phylogenetic trees, that was around when I wrote the initial natural limits, but it was in a very minimal stage. Now we've been able to make phylogenetic trees out of many different kinds of genes, many different kinds of proteins, and sometimes we can combine them all together. The problem is the phylogenetic tree you get for this protein is not the same as the other protein. It comes up with a different phylogenetic tree. When you try combining them together, trying to sort through what the actual tree is, you get a really, really low level of confidence. What does it really mean? Behe's book, Darwin Devolves, the idea that when we do see something new appearing, what it technically is the loss of something that already existed. And then last, other sources of information in the cell. Jonathan Wells did a chapter in the Theistic Evolution book that dictated how many things that we see operating in the cell do not owe their origin to a DNA sequence. Lots of other things are going on. Sometimes genes occur in pieces, and then the different pieces get arranged and rearranged in different ways. I think it's one gene in Drosophila fruit flies can account for thousands of different genes, different proteins from technically one gene. And many proteins have carbohydrate chains stuck on top of them. Well, there's a whole source of information in all of those as well. So I'll be looking into a lot of these other sources of information again to demonstrate that there are real natural limits to biological change. My professor at University of North Texas, Earl Zimmerman, I actually asked him to read this. And I just asked him, do you think evolution is represented fairly? And after reading, he said, yeah, you didn't get anything wrong about evolution. Because one of the big criticisms of any kind of anti-evolutionary material at that time, they don't really understand evolution. They don't get it. That's why I went to UNT in the first place. And so I want to make sure with my professor, because he was the evolution professor at UNT at the time, is it correct? And he was able to tell me, yeah, you basically got it right. So it was unusual at that period of time, comparing and contrasting different views, and showing again that there do appear to be limits to natural change. Well, I ended up being 9 minutes over. Questions?